morning, everyone. I will kick off our presentation. Thanks for joining us. My name is Angelique Dahlberg, and I am currently a PhD candidate at the University of Minnesota. I will be co-presenting with Diane Waller, a researcher with USGS and the lead investigator on this work. We have divided the presentation such that I will talk first, and then I'll hand it over to Diane to finish the presentation. And we will both be available to answer questions at the end. Also joining us today is Matt Barbour, another researcher with the USGS, who will also be here for questions. Our presentation will be on our work refining copper-based treatments that suppress zebra mussel populations and our main findings and progress to date. So before I get too far, I want to acknowledge that funding for this project was provided by the Minnesota Environment and Natural Resources Trust Fund, as recommended by the Minnesota Aquatic Invasive Species Research Center and the Legislative Citizen Commission on Minnesota Resources, as well as the Minnesota Aquatic Invasive Species Research Center, Hennepin County, the Pelican Lakes Association of Crow Wing County, the Fletcher Foundation, the Bay Lake Improvement Foundation, the Great Lakes Restoration Initiative, the U.S. Geological Survey Ecosystem Mission Area, and the Environmental Protection Agency. So we have a diverse set of supporters behind this work. We would also like to thank the organizations listed here on the right, especially Gabriel Jabbour and Wayzata Marine. And I'll add that we've had incredible support from a number of individuals and organizations, including some who have been instrumental in different parts of this work who may not be listed for today's presentation, but we're very grateful to everyone who has assisted us. A quick overview of our presentation. I will provide a brief background to give you some context for this research. And then I will talk about our research on veliger sensitivity to copper. At that point, I will turn the presentation over to Diane, who will share what we did this year on Lake Minnetonka, and then end with some upcoming plans. And as a disclaimer, although we will be discussing the effectiveness of a molluscicide, EarthTech QZ, this is not an endorsement. It's the product that we used. So before I talk about the research, let's talk about why we're doing this work. The animal that we're targeting is the zebra mussel. I suspect most of you are fairly familiar with this organism. Zebra mussels are small, two-shelled bivalves. On the screen, I have an adult in the upper left-hand corner. On the right, I have multiple adults attached to a native mussel. And then what I really want to draw your attention to is down in the bottom left is an image of veligers, or a stage of larval zebra mussels under magnification. And so while adults are relatively easy to find and identify, you can't actually see these juvenile stages unless you have a microscope. So zebra mussels were first introduced back in the 1980s. Since then, they've had some very profound impacts to native species and our native ecosystems. I won't get into any of that today. Basically, we know that they're a problem in our lakes and rivers, and the question that leaves us with is, what can we do about that? So What we are investigating is the use of copper to control and suppress some of these populations. Copper has a long history of use in aquatic systems. Historically, humans used copper on plating on boats to prevent barnacle attachment. Today, some paints used on boats still contain copper. But beginning in the 1920s, researchers and communities in Minnesota and Wisconsin started using copper sulfate to control algae in both lakes in Madison, Wisconsin, and in the Fairmont chain of lakes here in Minnesota. And today we've seen that use in lake management evolve to now include use targeting different aquatic invasive species, including zebra mussels. So this research that we will talk about today is a part of an ongoing effort to learn more about how copper controls zebra mussels. And to give you a reminder or an overview of where we're at today and how that ties into everything we've been doing, sort of rewind back to 2016. In 2016 and 2017, USGS and MASERC conducted a number of studies to better understand zebra mussel sensitivity to different molluscicides in different scenarios. In 2019, we partnered again to test the effectiveness of EarthTech QZ, which is a copper-based molluscicide, in St. Albans Bay in Lake Minnetonka. And if you've come to these showcases for several years, you've probably heard us present on that work. In 2021, last year, we tested predictive modeling to see if we could improve our treatment dosing ability. And we simultaneously expanded our understanding of non-target impacts from copper. And we did this in our mobile lab that we brought up to Pelican Lake. And that's what we presented on last year. So today I'm going to pick up where we left off last year and start by sharing our work looking at veliger sensitivity and how that can help us predict best copper concentrations to use in control situations. So I'll start kind of where we left off last year. So if you did see our talk last year, or even if you didn't, we spent time up at Pelican Lake in Crow Wing County testing some predictive modeling and working to expand non-target research. And the questions that we specifically sought to answer were the questions I have listed here, which are, can we accurately predict a concentration of copper that will reduce veliger numbers by doing bioavailability modeling? And using those predictions, what is the lowest concentration of copper that will reduce veliger numbers? And then are there non-target impacts associated with that concentration, and what are those? So as we work to answer those questions, we worked in a mobile lab in a setup that looked like this. Because this was last year's focus, and we're going to mainly talk about this year's focus, I won't really go into a ton of detail on this, but do please reach out if you want to know more or if you have any questions. Very briefly, though, we examined multiple organisms' sensitivities to copper. We had different tanks that were dosed with different concentrations of copper. And we studied the survival of zebra mussels, Daphnia, which are a large zooplankton, survival of a native mussel species, a snail species, and 3 fish species. And if you recall, we were also hoping to know not just adult zebra mussels, but how the villager life stage survived under these different concentrations. And I'll address that in a moment. We have some preliminary results from that work. They have not been peer reviewed, and so I'm not going to dive into this, but I just wanted to share. Here we're looking at the different organisms and their survival rate, 0 corresponding with no survival and 1 meaning complete survival. And our takeaway seems to be that our fish and our native mussel species largely were not sensitive to the concentrations of copper that we were using. However, the zebra mussels, the adult zebra mussels, and Daphnia and the snail species that we looked at looked like they probably or might be sensitive to some of our higher concentrations. So what about that villager life stage of zebra mussels? If you're familiar with this project, you'll know that unfortunately we did not get great data on our villager response. We have some information that we're working through, but we discovered partway through our trial that a lot of the villagers that were involved were probably dead before they reached our tanks. So we returned to this question this year, which is kind of where we pick up. So this year, once again, we collected villagers and we looked at their survival in different concentrations of copper in lake water. Our setup was a little different from last year. Instead of tanks, we used jars. Not a huge change, but worth noting. And our jars were dosed with 0, 20, 60, 100, 140, 180, and 220 micrograms per liter of copper. And micrograms per liter corresponds to parts per billion. So if that's an easier way for you to think about it, we looked at concentrations of 0 or no copper added, and then copper added either from 20 to 220 parts per billion. And we exposed the villagers for 24 hours. After 24 hours, we then took the villagers and placed them in untreated lake water for an additional 24 hours. And at the end of that second 24 hours, we assessed their survival. To assess the survival, we used microscopes. We pulled our samples from the jars, and looked at each villager that we had in there and determined if it was alive or dead. I had 3 incredible volunteers help me with this. And the image on the right of the screen, that circle, is what they spent the day looking at. And if you look at the middle of that circle near the intersection of the lines, I think you can see my mouse, we have a zebra mussel. And we don't have the results of this to share yet, but it's much more promising than last year. The villagers were not dead upon arrival. So we are working our way through these results. Stay tuned for more information on that. And with that, I'll hand the presentation over to Diane to share the rest of what we're working on.
Okay, thanks, Angelique. We've had a little excitement here this morning, so just made our way back in from a fire alarm. So I'm glad we made it back in the building. So I'm going to talk in the second half of our presentation. You want to go to the next slide, Angelique? About the open water component of this research. So from past years, you might remember that we investigated how effective a low dose of copper as Earth Tech QZ would be for suppressing villager settlement. This phase 1 occurred in Lake Minnetonka. We used St. Albans Bay as our treated bay and Robinson's Bay as the reference. So we've been monitoring both bays for 2 years and then added a third year of monitoring in St. Albans this past year. Next slide. So the next several slides will be an overview of some of the trends that we've seen in those 2 years of monitoring. This graph shows the number of juvenile mussels that have been found on settlement plates in August and October of each year from 2019 through 2021. So the reference bay or Robinson's is shown in blue and St. Albans, a treated bay, in red. You might notice that the number of mussels in August is always higher than in October, primarily because as mussels grow, the available area on the plates is reduced. So notice in 2019, we had almost no settlement after treatment in St. Albans. We did see an increase in 2020, but it was still well below that of the reference bay. In 2021, settlement in August was still lower in the treated bay than in the reference, but this red bar does represent still tens of thousands of juvenile mussels. And by October, the number of mussels on samplers in the 2 bays was pretty similar. So overall, it looks like the treatment successfully reduced settlement in the first 2 reproductive seasons. And then by the second or the third season, settlement was reoccurring. And St. Albans has a lot of boating traffic. It's isolated from the main part of Minnetonka. So it's really not surprising to see this upward trend 2 years after treatment. Next slide. So there's a lot on this slide to illustrate what one of our main interests here is how Native communities are responding to the treatment and if we can see any trends related to the decline of zebra mussels after a treatment. So just starting with the 2 graphs left and right, this is for 2 of those native groups, benthic invertebrates and zooplankton. The graphs are illustrating mean abundance with the error bars representing 1 standard deviation. Now in these graphs, the reference or control bay is shown in red and the treated bay is shown in blue. So starting with the graph on the left, benthic invertebrate abundance was initially lower in St. Albans or our treated bay than in Robinson's. And that was primarily because of differences in the substrate and habitat type between the bays. So we aren't directly comparing the reference and treated bays, but are looking at trends over time within a bay. So you can see abundance declined slightly after treatment in 2019, but this was not statistically significant. In 2020, abundance of the benthic inverts was similar to what it was at pre-treatment. And in 2021, the abundance was down early in the summer sample, and then it bounced back up later in the summer. And you'll notice that that trend was actually reversed in the control or reference bay. Over on the right-hand side of the screen is the zooplankton abundance. In 2019, you can see immediately after a treatment, it was significantly reduced, but did show some indication of recovery in the 14-day post-treatment sample. In 2020, abundance was above that before treatment. It then returned to pre-treatment levels in 2021. And really, we're wondering if the increase in 2020 might have been related to the decrease in zebra mussel population that year and perhaps less competition. So in addition to abundance and also diversity, which I haven't shown here, we're looking at changes in community composition over time. So non-metric multidimensional scaling is a way to show similarities between samples when you have complex data. So an example of zooplankton composition in St. Albans or the treated bay is shown in the middle graph. So there's a lot of information here. Just keep in mind, the greater the distance between two circles, the less similar the communities are. So the most obvious separations that we see are between the pre-treatment community, which is shown in the red circles, from though the communities 1 day after treatment, which is shown in the gold triangles, and then 14 days post-treatment, which are the green squares. So again, this shows that not only was there a decrease in abundance after treatment, but in fact, the composition of the zooplankton community also changed. In the first year post-treatment, those are shown in the green cross, and then a little later in the summer, the blue squares. And then 2 years, which are shown with the purple and pink asterisk, the samples clustered more closely to pre-treatment, the pre-treatment communities. So this seems to indicate that there was recovery 1 and 2 years later. We'll be doing a similar analysis for the benthic invertebrate community data, and we'll be adding our 2022 data to this when it's available. Next slide. So a concern with copper-based pesticides is their permanence in the environment. So we also want to know what concentrations are well after the treatment is over. So this slide is showing the copper concentration on the left-hand side right before the treatment, the very first point there. You see pretty minimal levels of copper in the water. Then during that 10-day treatment, it hovered between about 60 and 100 parts per billion, which was our target. And then post-treatment, on the right-hand side, you can see in 2019, by 60 days after the last treatment period, it had fallen back down to kind of pre-treatment levels. But what's interesting on this graph is we've continued to follow copper concentration in the water at various sampling points, and I apologize, the 2020 data are not on here, but to point out that the circled concentrations in 2021 are up around 60 parts per billion in both our treated and reference bay. So it really appears that other copper-containing pesticides are being applied in these bays, and it somewhat confounds our interpretation of what impact these low-dose treatments might be having. So we'll finish up our long-term monitoring in St. Albans next month, and then complete the final analysis of our zooplankton, macroinvertebrate, and zebra mussel data. Next slide. So phase 1 of this research did show some success with a low-dose copper treatment. But as Angelique mentioned, we really want to further refine how we're applying copper. So phase 2 of this research started in 2021, and it differs from this first phase that was started in 2019 in several ways. We selected 2 different bays, again in Lake Minnetonka, this time North Arm and Maxwell Bays, both in the kind of northwest portion of the lake. Both have very well-established zebra mussel populations. Maxwell Bay was selected for the treatment, and it is about twice the size of St. Albans Bay, so we're looking at a larger body of water. We identified areas within North Arm and Maxwell that were similar in substrate and vegetation, so they're more comparable reference and treatment sites than we had for St. Albans and Robinson's. Based on the work in 2021 and doing some BLM modeling, we lowered the target copper concentration from 60, that was the target in St. Albans, to 45 to 50 parts per billion. Also, the application in St. Albans was to the whole bay, but shoreline treatments are commonly used to limit the application to 50% of a water body as specified on the Earth Tech label. So this is the approach that we used in Maxwell Bay. And very importantly, we conducted more extensive pre-treatment and post-treatment monitoring of the native communities for this phase of the project. Next. Our pre-treatment and post-treatment monitoring included, of course, zebra mussels. So we're looking at veliger density, adult density, and also mortality of caged adults during and after the treatment. And then, of course, juvenile settlement on samplers. Volunteer network of residents along Maxwell Bay is helping us in this effort by placing samplers under their docks. For native communities, we've added phytoplankton abundance and composition, along with zooplankton, macroinvertebrate settlement on Hestradendes samplers. We're also looking at native juvenile mussel survival and growth right after and a longer term after the treatment. And measuring, of course, copper concentration, not only in the water, but also in the sediment and biota. So we monitored veliger density this year every 2 weeks, starting in June and then weekly in July, and scheduled the first copper treatment to coincide with peak water temperature and veliger density. So the first treatment was applied July 20th. It was repeated 3 times every 4 days, with the last treatment on August 1st. On the center figure there, you see Maxwell Bay, and the shaded portion is our application path, which was about 75 meters from shore. The box Bs that you see represent sampling stations. We had 7 of them around Maxwell Bay and then 3 of them in North Arm Bay. And this is where we collected plankton toes, put Hestradendes samplers, and placed our caged zebra mussels and native mussels, and also collected water and sediment samples. You also see 3 transect lines across the bay. This is where we took additional measurements of copper concentration, and then also measured copper at the channels to Crystal and North Arm Bay. Next slide. So there's a lot of information on this slide. It's illustrating copper concentration in surface and bottom water samples at 6 of those locations during the treatment period. So let me just highlight a few of the main points here. First of all, at sites within the main bay, the copper concentration was near target, and you'll see a midpoint at the center of the bay where we also achieved that 50 parts per billion, which really indicates that the copper dispersed across the bay. Second, there was vertical mixing to a different degree depending on the depth. So there at the midpoint, it's a little deeper. So the copper concentration near the bottom wasn't quite as high. Whereas if you look at the upper left-hand graph, this is a shallower area of the bay, and we had pretty even distribution of the copper in those shallower areas. The third thing I want to point out here is we were out there on some really windy days in July doing these applications, and the wind speed and intensity explained a lot of the variation that we saw across the bay. You'll notice the site down in the lower right-hand corner. This is in the southeast corner of the bay, where we had a strong northwest wind. So it was blowing the copper across the bay. And I guess the good news is that the veligers were being blown with that copper to that higher concentration. And lastly, if I can point out this small finger part of Maxwell Bay. In this area, in our first treatment, we were significantly above our target for copper in the bay. We then adjusted our application method in later treatments to try to bring that back down in line with the target of 50 parts per billion. But because of less water movement in this area, the concentration was still higher than most other areas in the bay. Next slide. After treatment, we collected samples 1, 7, 14, and 30 days, and we'll be collecting our last one 60 days post-treatment in October. So we have a lot of samples yet to work through, and I can just give you some very preliminary results. First, we saw the low dose treatment killed about 50% of caged adult mussels in the main bay, and 100% of them in that finger part of the bay where the concentration was quite a bit higher. We pulled September settlement plates a couple weeks ago. They have not been enumerated, but we did observe a small number of juvenile mussels on the plates in Maxwell Bay, while in comparison, those in North Arm, the plates were almost completely covered with juvenile mussels. Native mussels at the 2 sites in the Little Finger did have high mortality in response to that extra high copper, but out in the main part of the bay, we had nearly 100% survival of juvenile natives. We're also going to compare growth between the treated and reference bay to see if there were some non-lethal effects of copper on them. So our zooplankton, phytoplankton, and macroinvertebrate samples are still waiting analysis, but I will just point out that our Hesterdendes that we collected from North Arm, the reference bay, in our last sampling period, were about 90% covered with zebra mussels and not much of anything else. Next slide. So this project will continue for 2 more years, kind of a phase 3. And in this next phase of it, we're going to continue post-treatment sampling to determine what level of settlement suppression was achieved with the shoreline application at a lower concentration. See what that looks like in 2023. And also then whether we can see a benefit to native communities if there is a reduction in zebra mussels, or what risk that copper application might have posed to them. The culmination of all this work on low dose copper is going to be a structured decision-making workshop with stakeholders in 2024. So the workshop will be an opportunity for us to assemble all of the science available on open water control actions for invasive mussels, and then bring together stakeholders to look at developing a strategy for how established populations of zebra mussels might be managed in Minnesota lakes. And with that, I'll move to the last slide and say thanks for your attention. And Angelique and Matt and I are happy to take questions.
Thank you, Diane. We have a question here that says, how does copper affect other species in the treated lake?
Yeah, so that is one of our main questions. Well, I think maybe I'll let Angelique talk a little bit about some of the work that she did last year in the trailer, because that was primarily looking at non-target species.
Yeah, I am scrolling back to this slide, which I didn't really go into a whole lot of detail on, but we last year looked at the species that I have presented up here, adult zebra mussels, Daphnia, which are the large zooplankton. Fat
I think you have to share your screen again, sorry.
Oh, I have to share it again. Thank you. It says it's sharing, all right. Yeah, it was weird on my end. Thank you. So I'm gonna go back to the slide, and these were the different species that we had in our mobile lab last summer that I just kind of mentioned very briefly in the presentation. But we looked at adult zebra mussels. We looked at Daphnia, which are that large zooplankton species, fat mussels, which are a native mussel species, banded mystery snails, largemouth bass, bluegill, and fathead minnow. And the concentrations that we looked at were 10 parts per billion, 25, and 40 parts per billion. And these results are still preliminary, but what I mentioned earlier was that at these concentrations, we don't see a real difference between the untreated controls and the different treated tanks for our fish species or for our fat mucket mussels. We do see a little bit, if you can follow the mouse on the screen, we see there might be some sensitivity and survival among zebra mussels, among Daphnia, and among banded mystery snails. And really what we're looking for here is this difference between the control and these treatments. Obviously, this is only like a small subset of species that you would find in a lake. There's a huge body of literature out there on how different organisms respond to different formulations of copper. We're actually doing a large literature review of many of those studies. Initially, there were like 7,000 studies that came up, and we've narrowed it down to just under 2,000, and we're still working our way. Turn on the camera. We're still working our way through, but yeah, I don't know. I mean, every organism is a little different. The response is different in different water bodies. This was kind of the question behind our predictive modeling was, well, we know copper toxicity changes with bioavailability, which is dependent on water chemistry, and every lake's going to have different water chemistry. So a zebra mussel could be different or could be sensitive to different concentrations in different waters. But I think I've really gone overboard in my answer, but hopefully this figure kind of lets you know what some of our major organism breakdowns could look like.
Thanks, Angelique. Yeah, we've got a question from Amanda here. From the results, do you believe copper can be a successful treatment option? Since zooplankton are already impacted by the presence of zebra mussels, will the decrease of zebra mussels be better than any potential lost zooplankton or invertebrates?
That is the million-dollar question. So yeah, that's what that is really a main focus of this second phase of this work. We really increased the frequency of sampling for zooplankton and benthic invertebrates. It will be not an easy question to answer because there's so much variation in zooplankton populations, but we are hoping that we'll be able to kind of tease some of that information out.
Thanks so much for answering that, Diane. And also, there are several answered questions. Another researcher, Matthew, has been answering them. typing them in. So if everyone wants to hear the answers to that, you can certainly click in there. We have one more question here from Ian. Has the treatment affected any of the native plants or soil qualities?
Yeah, that was not that was not within the scope of our work, just because, you know, there was only so much that we could include in our monitoring. We have been kind of keeping track of, so to speak, what the vegetation looks like around our buoy locations in particular, but we were just limited in what we were going to be able to monitor. So in this particular phase of the project, we did not have a component to quantify effects on vegetation.
As far as the soil goes, if you mean like the substrate of the lake, we did collect sediment samples in Maxwell Bay in this most recent treatment, but we have yet to analyze those samples yet, so it's yet to be determined. But it's something we are going to look at.
Thank you for that addition, Matthew. If anyone has any other questions, you can certainly enter them at this time. In the meantime, I'd like to thank all the presenters that shared today on their research on zebra mussels and copper. Oh, we have one more question. What is the fate of copper in the lake systems over time?
So I can answer this one. Copper absorbs or attaches to different organisms or particles in the water pretty quickly in the grand scheme of things, and ultimately makes its way down to the bottom of the lake, and typically is just kind of sequestered there and usually remains trapped in the bottom sediments there indefinitely, usually.
Thanks, Angelique, for that answer. Unless we have any more questions, that can be our last one. And I'd like to again thank the researchers for presenting here today.